So I'm excited to be here talking to you about how we think about and study disease at the molecular level. Fundamentally, studying disease is just the process of taking something that isn't working the way that we expect and trying to figure out why that is. And this is a type of problem solving that most people do all the time, often without even realizing it. So for example, say you got up this morning and you were getting ready to go to work, and you went out to your car, getting ready to go, and then your car doesn't start. This is something that now isn't working exactly the way that you expect. So this will likely immediately kick off a whole set of problem-solving steps that you undertake naturally. So for example, you might check and make sure that you have enough gas, the temperature is okay, none of the gauges are giving you a warning light or anything. And if everything looks okay at this level, so externally everything seems fine, then you might go a step further and say pop the hood to get a look at some of the smaller parts that work together to make your car run within the engine. And this is exactly what we're doing when we study disease, except for instead of looking at something like our timing belt, instead we're looking at your cells. And now if you're like me, every time you see this kind of a figure or a diagram like this, you're probably wondering, how do we get from this cell to a whole human person that I care about when I'm thinking about disease? It seems like a pretty big leap. And the answer, at least in part, is that not all of our cells look like this. We actually have a whole wide variety of cells that have a whole bunch of different shapes and are carrying out a lot of different functions all the time. So for example, we have specialized cells in our brain that help us think. We have specialized cells in our gut that help us digest food. We have specialized muscle cells that help us to lift things and move around. And we have specialized skin cells that keep us protected from environmental harm like the sun or other damaging things that we might encounter in day-to-day life. And now, one thing about these cells is that even though all of them look very different and that they're all carrying out very different functions, is that they actually all have DNA as an instruction manual. So all of them have this in common. And this DNA instruction manual is what tells each cell how to be itself and how to carry out all of the functions that it needs to on a day-to-day basis. Now, as you might imagine, for an instruction manual having to give instructions for such a wide variety of things, it's pretty big. And in fact, if we typed out the instruction manual in our DNA into an actual book, it would be over 300 feet tall. So this is an instruction manual that's actually bigger than the Statue of Liberty and only slightly smaller than the Washington Monument. So this is a lot of instructions. But to me, this isn't even the most remarkable thing that our cells are doing. One of the things that I think is the most impressive is that each of these cells, with their different forms and functions, actually is working from the exact same instruction manual. So whether you're a brain cell or a muscle cell, you have the same set of instructions. So you might imagine that there's a lot of extra information in here that, say, is telling you how to be a brain cell that the muscle cell doesn't necessarily need. And so one question is, how do we get from this common instruction manual to all of these really different forms and functions, even though they have all this unnecessary extra information? Some extra information in an instruction manual is a problem you may have already encountered when you were trying to figure out why your car wasn't starting. So for example, anytime I need to visit my car manual, even if I have a very simple question like, how does my air conditioning unit work? When I flip to that section of my car manual, what I find is this. It's a bunch of instructions for 3 whole different groups of cars, all the way from the top of the line model down to what I have, which is a bunch of extra information that I don't really need when all I want to know is a simple thing about my air conditioning unit. And this can feel a lot like having to wade through an incredible amount of information that I'm never going to need. And if this has ever been something that's frustrating to you, and I know that it's been frustrating to me, you can spare a thought for our poor cells who have an instruction manual that's bigger than the size of the Statue of Liberty. So they've actually done something that I think is quite clever to deal with this problem of having to wade through all of this, which is that instead of going through this big, huge instruction manual all the time, each cell actually works from copies. And they only make copies of the parts that they need. And so as a result, instead of having to work from this very large instruction manual, most of our cells are actually working from a bunch of smaller booklets or instructional copies of the DNA. And they do this through a specialized molecule known as RNA, which allows them to copy the DNA and then work from there. Well, so what are these instructions for? Well, in most cases, they're to make something called protein. Now, when I hear protein, the first thing that I always think of is food, because often I'm mostly thinking of food. But it turns out that the protein that we think about when we eat is very similar, or it's related to the kinds of protein that we're talking about at the cellular level. So the food that we eat actually gets broken down into the building blocks for proteins that our cells then reassemble into the kinds of proteins that they need on a daily basis. And the kinds of proteins that they need are basically small, tiny molecular machines that are all working together as though they're on a factory floor, each carrying out specialized individual tasks that together make the cell happy and healthy. And this includes a really wide variety of tasks. So for example, there are some proteins that act kind of like tow trucks, where they're towing other proteins around where they're needed in the cell. There are some proteins that act like the photocopiers, so they make the RNA booklets from the DNA instruction manual. There are even some that act sort of like trash compactors. They go around the cell clearing waste and keeping everything clean and running smoothly. And this flow of information from the DNA instruction manual to the RNA booklets to the final product of protein is such a fundamental, important flow of information in life that we actually call it the central dogma. And it's from this central dogma that we're able to get this really wide variety of specialized cell types, all from the same instruction manual. Now at this point you might be thinking, I came here to hear a talk about disease, why are you telling me a basic biology lesson? And that is a totally fair and understandable point. But part of the reason that I like to start here is just to highlight that when we're thinking about disease, even at this very small level of a single cell, there are a lot of levels where things can go wrong. And we have to think about all of them when we're trying to figure out what's going wrong during the course of disease. And so this can be kind of overwhelming. We need to decide where to begin. We need to know which levels to start looking at to figure out what's going wrong. And so one good place to start looking is actually just at the instruction manual itself. I told you it's common in all cells, so this seems like a reasonable place to start. Additionally, very small changes at the DNA level can make a really big difference. So for example, if we again revisit our car manual, say I'm interested in changing my oil, I flip to this section, I see, okay, checking the oil level, this is important, this is what I'm interested in. If I make a very small change, say inserting a single letter, now all of a sudden it says checking the soil level. This now doesn't really make very much sense. It's not very helpful for the task that I'm trying to carry out, which is checking my oil level. And the same kind of thing can happen in your DNA. And it turns out there are several categories of this kind of change or mutation that can be happening, and this is only one of them. So for example, adding a letter is a type of change that we call an insertion. You can also change a letter, so instead of checking the oil level, now I would say checking the oil lever. You can also make a deletion, where you've removed a letter, and now it says checking the oil level. And all of these can change the meaning of the sentence. And the same thing is happening in your DNA all the time, except for instead of having all of the letters of the alphabet, our DNA is actually made up of only 4 letters, known as nucleotides, but the same principles apply. So in this example, we've changed a T to a C, but the exact same effect can be the case. The meaning of this segment can be changed just by a simple 1-letter substitution. So it's obviously very important for us to see what's in the instruction manual and what the DNA says. But that can be sort of challenging. How do we read the instruction manual? Now our cells, have evolved many proteins or little molecular machines that do this for them all the time. But we had to build machines for ourselves to do this if we were interested in reading what the DNA instruction manual says. And so to do that, we designed these machines that essentially what we can do is we can take the DNA, chop it up into little bits, then put them on these machines. They will read the DNA sequence letter by letter, and then we can assemble it again later to see what it said altogether. But I want to highlight that just knowing what the DNA says isn't quite enough. So these sentences, checking the soil level, checking the oil lever, out of context, these might make plenty of sense. There's definitely occasions where I might want to know what my soil level is. I'm sure there are some oil levers somewhere. It's not enough just to read the sentences. We have to know what they were meant to say so we can tell if they're saying something different. And so the way that we do this is we look at populations of people, and we look at the DNA sequences from people who are unaffected by disease, and then we compare them to the DNA sequences of people who are affected by disease. And when we do this, we can see that, say, everyone who's unaffected, their sentence might say checking the oil level, whereas people who are affected may have different variations of these changes in that sentence. And this tells us that the problem in this particular instruction manual is with this sentence and not with any other part of the book. Now, this type of analysis is obviously more powerful the more and more sequences you have to compare here, because then you can be more and more confident about which sentence in the book it is that's causing the problem. But initially, this wasn't very feasible or practical because sequencing the human genome was very expensive and it took a really long time. So sequencing a single human genome for the very first time took 15 years and it cost $3 billion. So this obviously isn't something that you can do if you need thousands and thousands of sequences to study a disease. But fortunately, sequencing technology has not only gotten much faster, it's also gotten much cheaper. So from $3 billion when we sequenced the first genome, today it costs about $1,000 to sequence a single human genome. And instead of 15 years, we can do it in about an hour. And so the result has been that we've been able to sequence more and more human genomes, and this has given us more and more power to study specific diseases. And to highlight how this has been helping us, I'm going to just focus on one specific group of genetic diseases, known as trinucleotide repeat diseases. And don't worry if you don't know what that means right away, I'm going to explain it in just a few moments. And so within this group of trinucleotide repeat diseases, I'm going to specifically talk about two, Huntington's disease and muscular dystrophy. Now, one of the things that I think is interesting about these diseases is that even though they're in the same family, they affect very different tissues. So Huntington's disease primarily affects our brain, whereas muscular dystrophy, not surprisingly, primarily affects our muscle. And more specifically, Huntington's disease is a fatal genetic disorder. It affects tens of thousands of Americans. And what it is fundamentally is a progressive death of brain cells. So you have fewer and fewer of them over time. And we can see this manifests in essentially holes that appear in the brain as your brain cells continue to die. Now conversely, muscular dystrophy is actually a group of inherited disorders. So there's a lot of things that fall under the umbrella of muscular dystrophy. And so this impacts hundreds of thousands of people worldwide. And instead of affecting your brain, this is a progressive wasting away of your muscle tissue. So all of us, are born with, over our lives, we have a certain amount of muscle tissue, and this is what we move forward with. But if you have muscular dystrophy, whatever amount you have, it basically gets less and less over time, until eventually you have trouble, moving around or lifting things, and it can manifest in very specific deformities as it progresses along. Now, as you can see, these affect very different things, but I told you they are in the same family of disease. So what is this? What is the family of trinucleotide repeat diseases? What is it that they have in common? Well, trinucleotide repeat diseases are actually are caused by a very specific kind of mutation. And it's actually a very specific kind of insertion mutation. Where we've been talking about an insertion of a single letter, in this case of trinucleotide repeat diseases, it's the insertion of 3 letters over and over and over again. So instead of checking the oil level, we now instead have checking the oil level, and it's much longer. And this can have different effects to the cell depending on where this occurs. So for example, in the case of Huntington's disease, the extra sequence causes problems at the protein level. And so we have these extra instructions, and the way that they manifest is by basically making instructions to make too many hooks, say, on this tow truck protein. And that might not seem like such a big deal, but if you have way too many hooks on your tow truck protein, the end effect is that they get tangled up together. You have all of these proteins that have these hooks flying about, and this is a problem that only gets worse over time as you have more and more of these proteins with too many hooks that are getting tangled up. And this is why this is a disease that gets progressively worse over time. Now conversely, in the case of one specific type of muscular dystrophy known as myotonic dystrophy, the problem with the extra sequence happens at the RNA level, so at the instructional booklet stage. And here, when we have a bunch of extra instructions that get copied into the booklet, we get a much longer booklet. And the reason that ends up causing problems is because, say, this booklet was ordinarily supposed to bind and interact with one protein. Now it's way longer, there's a lot more pages, and so instead it binds to way too many proteins. And in this way, it acts more like a sponge, where it's soaking up these proteins, and as a result, they're not able to do the job that they were supposed to in the rest of the cell. And so this is how, even though these two diseases affect very different tissues and look very different on the outside to us, they actually have a very similar cause at the DNA level. And this is something that we wouldn't necessarily have known if we hadn't been able to sequence the instruction manual or look at the DNA itself. And so with that, I just want to highlight that cells have a really tricky task to carry out. Not only are they all working from the same instruction manual, but they also have a whole variety of complicated processes that they have to be doing all the time. And for the most part, they do a very good job of this. Secondly, even very distinct diseases can have surprisingly similar causes that would be very hard to detect if we didn't zoom in, pop the hood, and look at the cellular level. But finally, I've talked a lot about identifying diseases and how we figure out what's causing them. But ultimately, that's not really all we want to do. We want to be able to study them and figure out how to treat them. And so one way to do that is using model organisms, and that's something that Sam is going to talk to you about